Good afternoon. I'm Jayathilaka from Center for Dengue Research, University of Jayathilaka. So today I'm going to present our findings on D614G mutation associated with higher transmission in Sri Lankan SARS-CoV-2 virus strain. So as all of you know, since March 2020, Sri Lanka had several confined COVID-19 clusters coming up in several parts of the country. And since in October, we are having this current epidemic outbreak. So SARS-CoV-2 virus carrying D614G mutation is rapidly circulating globally. So therefore, we carried out a mutational analysis to investigate the D614G mutation in SARS-CoV-2 virus strain circulating in Sri Lanka. So first, what is this D614G mutation? It is that at the 614th amino acid position of the spike protein, the amino acid aspartate is, which is shorthand as D, is replaced by glycine, which is shorthand as G. So the virologists call this mutation as D614G mutation. So we have done sequencing of 35 SARS-CoV-2 real-time RT-PCR positive samples, and since the beginning of this pandemic. So as per methodology, we carried out sequencing in two different approaches. Initially, this shotgun metagenomic approach, and then later targeted sequencing approach, which we thought better than this shotgun metagenomic approach. So, so at the end of sequencing reads by both methods, and also after further analysis, the resulting sequences were compared against this annotated SARS-CoV-2 isolate reference sequence from Wuhan for mutation. So we observed that out of 35 virus strains that we sequenced, 29 had this D614G mutation. So, and interestingly, out of 23 strains that we sequenced from this current outbreak, 22 had the mutation. So, so that means from October to November, out of 23, 22 had the D614G mutation. So if I go back to the beginning of this this pandemic, in March, the virus strains of several returnees from Europe had this D614G mutation, and later, but at that time, the returnees from Bangladesh or Middle East did not have this mutation. And then later in April, we observed this mutation in community clusters seen in Colombo municipal area. And also then in May, we observed this mutation was associated with the Navy cluster, and also in July with Kandakadu Drug Rehabilitation Center cluster. So therefore, as seen globally from March to November, this virus strain with D614G mutation became the predominantly circulating virus strain in Sri Lanka. So this D614G strain is actually shown to associated with higher viral loads, which means that if the person is infected with this virus strain with this mutation, the patient tend to shed higher viral load compared to patient infected with this previous virus strain. So therefore, according to our, and therefore it has higher transmissibility as well. Therefore, according to our findings, virus strains carrying D614G mutation is likely to have displaced the previous strain, which has less transmissibility, and also it might have caused this current outbreak. Thank you. And also, I'm really grateful for our funding agencies, CDR, WHO for University of Jayathilaka, British High Commission as well. Thank you very much.